In this video, we are going to go over different strategies for antibody structure prediction and the domains of applicability for each. The fundamental problem of antibody structure prediction is going from a one-dimensional sequence to a three-dimensional structure. The classical approach to this problem has been template-based homology modeling, in which a sequence is grafted onto the structure of a close homologue. Starting with AlphaFold in 2018, a new generation of machine learning algorithms for protein structure prediction has emerged. Some are more general models that are meant to cover all biomolecules and small molecules, and others are more specialized for a particular domain, such as antibody-antigen systems. If we look at Schrödinger's antibody structure prediction workflow, which relies on template-based homology modeling, it all starts with a simple sequence alignment to identify frameworks that are highly similar to the query sequence. It is worth keeping in mind, though, that framework regions are notoriously easier to predict than loops. Next, a curated antibody loop database is filtered based on sequence similarity and length, as well as its ability to be grafted onto the chosen framework. The remaining loops are then clustered and ranked based on cluster size. A representative of the largest cluster is then chosen as the representative for that loop. An initial homology model of the framework is then made, and the CDR loops are grafted onto it. And finally, Schrödinger's prime tool is used to repack the side chains and minimizes all atoms of CDR loop residues plus other non-conserved residues. This workflow is particularly useful when high homology loop templates are available. Additionally, since the ML models are trained on public training data, template-based models are the best way to use proprietary in-house models as templates for predicting homologous structures. While there are many machine learning workflows available, many of whom share the same characteristics, Schrödinger offers a modified version of the immune builder workflow from the Dean Group. The workflow begins with A, bodybuilder 2, generating an ensemble of 4 structures. The structures are then aligned, and the one that is closest to the average is chosen. We then add in prime minimization as a final step to refine the side chains. While most ML models are evaluated based on C-alpha RMSD, for downstream modeling tasks, the proper placement of the side chains is essential. This machine learning-based workflow can be run at a much higher throughput than the template-based method and is recommended whenever you are looking to predict the structure for a larger library of sequences. It can also be a good alternative for when you can find only low homology loop templates. Even though no high-quality template exists in the training data, one of the hopes when using a machine learning model would be for it to be able to extrapolate beyond the information that is available in any single template. The degree to which that extrapolation beyond the training data is possible with current models is unclear. If we take away the CDR-H3 loop, both template-based homology models and ML models have been able to consistently predict high-quality structures. In this histogram, we look at the variability in CDR-H3 loop length in a particular antibody database. The high variability in length and sequence identity makes the H3 loops difficult to predict using template-based methods, since it is unlikely that you will find a loop template sequence with high identity, as well as for machine learning methods, since there are still limitations in their ability to extrapolate. In fact, a 2023 benchmark of different machine learning methods tried to characterize the per-loop performance. As you can see, the performance for H1, H2, L1, L2, and L3 was exceptional across the board. However, they all appear to be consistently struggling with the H3 loop, with average RMSDs of roughly 3 angstroms. There is reason to believe that advances in antibody structure prediction algorithms will likely lead to an improvement in H3 loop prediction, but it is certainly not solved currently and is a very difficult task given the diversity of the data. To address this fundamental problem of H3 loop diversity, Schrödinger developed a de novo loop prediction algorithm that, quote, samples the loop conformational space iteratively with varying focus and strategy, using the energy function as a guide to locate and identify low-energy loop candidates. This is a much longer prediction than any of the other ones previously discussed, but has been shown through various benchmarking assessments to be a top-of-the-class tool for H3 loop refinement. If you are using a template-based homology model, we would suggest using this antibody loop modeling workflow if the template H3 loop similarity is below 40%.